Welcome to the second part of the AI in chemistry and biology lecture. Last time we talked about sequence-based models, then also protein structure prediction, and after that, structure-based models. And today, we will mainly talk about the generative models for de novo design of proteins and small molecules. But before that, I would like to make a small excursion and talk about the challenges associated with training machine learning models on biological data. And that is what we will start with right after like a short recap of what we've learned last week, because many of these basic things will be important again today. So we have seen that proteins are chains of amino acids. And there are these 20 standard amino acids that all proteins are built from. And all these amino acids, they have a backbone. So that is this triangle of N, C, alpha, and C atoms here that are common to all amino acids. And then what differentiates the 20 amino acids from each other is what is attached here at the C alpha atom, which is called the side chain that gives the amino acid its specific properties. And then small molecules, we've seen that these are just organic compounds that are relatively small. So their structure usually allows to synthesize them quite easily in a chemical lab. And most drugs are small molecules because they can enter cells, bind to proteins, and so affect some biological processes. And so the chemical structure and the composition of small molecules defines its function, and therefore, if you engineer small molecules, you change these to achieve some specific interaction patterns with desired proteins. And we've looked at AlphaFold 2 to fold protein sequences into their native structures. And we've seen that after taking this protein sequence that you would like to fold, AlphaFold enters this genetic database and makes a multiple sequence alignment. And then it also checks a structure database for structures of related proteins that might already exist. And then it takes a guess of the first version of the pairwise distances between all amino acids in the protein using these templates. And then all this information is processed by the main architecture of AlphaFold into a protein structure that is then also refined using this recycling strategy. And we've also seen that because proteins rarely act alone, but usually interact with other small molecules or DNA or RNA, the recent folding models, they also include other biomolecules, such as like metal ions and small molecules and DNA, and then predict immediately like the structure of the whole complex. So you give them a sequence and maybe also Smiles code of a small molecule, and then they might predict firstly this structure of the protein, but also at the same time the binding conformation of this HIV protease inhibitor to this enzyme. And then we've looked at graph convolutional neural networks, that these are very well suited to model unordered data, such as social networks or molecules. And these GNNs, they transform the embeddings that are stored in the nodes of the graph and the edges of the graph, maybe also a global context vector through message passing. And then the resulting embeddings in these structures include information about the connectivity and the neighborhoods of the nodes and edges. And we've also seen that these GNNs can be useful to model molecules and proteins. So they can be used for the prediction of properties of small molecules, like that model that predicted antibacterial activity. They can be used for protein function prediction, or also, as we have seen, for the prediction of binding affinity between proteins and ligands. All right, I hope that refreshes the most important things we've seen last week. And now I would like to transition to the challenges that we face when training machine learning models on biological data, and then also show some examples of models that emphasize these challenges. So biological data commonly comes from databases that are based on community contributions. So that could be UniProt is a database of protein sequences and the functional annotations. The protein data bank, you've heard that a couple of times, is a database of protein and other macromolecular structures. And then PubChem would be another chemical database, but these are built on like the contributions of thousands of researchers that deposit their structures and experimental results in these databases. And this brings a couple of problems. Like, first of all, data is still scarce. So high quality labeled biological data is not so easy to find. So the protein data bank, for example, contains about 250,000 structures. But then if you want to predict binding affinity, or train a model to predict binding affinities between proteins and small molecules, then the data set of labeled complexes is about 20,000 or so. And then the data quality and the noise levels are a problem because these biological data sets often contain high noise levels because of variability in the experimental conditions and assays that people use to generate this data. And there are also often missing values, measurement errors, or just incomplete annotations. And there's also a lot of data imbalance present in these data sets because there's an overrepresentation of certain classes, or for example, well-studied proteins, such as the HIV protease that we've seen before, and an underrepresentation of other less interesting proteins. So the first problem I would like to talk about is this bias and redundancy problems. So as I've mentioned, these data sets may be biased towards well-studied proteins and contain a lot of redundancies because of that, because many researchers deposit structures of the same kind. And these redundancies are very often not really recognized because the data type is so complex. So it's not so trivial to find that two structures like this are actually show the identical interaction patterns between this small molecule and this protein. So people frequently use like wrong proxies for similarity, such as using just the protein sequence identity as a measure of protein ligand complex similarity. And that doesn't work because complexes with a low sequence identity, so that share only a small part of their sequence, can still have identical interaction patterns between proteins and ligands. And then models are usually or very often trained on these highly redundant but not very diverse data sets, often with like undetected overlap between the training and the testing data. And an example for that would be in this task of structure-based binding affinity prediction. So where you predict the interaction strength of a protein and a small molecule from their three-dimensional interaction structure. And the main data set that is used for that is this PDB-bind database that collects all these complexes from the protein data bank and assigns searches affinity labels for it. And this contains a lot of redundancies arising from multiple experiments on the same protein. And then usually this CASF, so comparative assessment of scoring functions, benchmark data set is used to compare different models and assess generalization. And this was initially designed to benchmark classical physics-based scoring algorithms. And it was sampled from like sequence similarity clusters in this PDB-bind. And there are more than 20 peer-reviewed and published models that train on this train and test evaluation setup. And there have always been these like data leakage concerns in this field. So we decided to look a bit more closely into that. And we computed 3 pairwise metrics for the entire database, including the CASF benchmark. So we looked at the ligand similarity. This is the first column here, which is basically just a Tanimoto similarity between count-based molecular fingerprints of the ligands. We looked at protein similarity, which uses like TM-Align, a tool to find the optimal alignment of the 3D structures of two proteins and outputting a score between 0 and 1. And then finally, we also looked at the binding conformation. So we computed an RMSD between all the ligand atoms after pocket alignment using TM-Align. And this resulted in 3 pairwise similarity matrices, each based on about 190 million comparisons. And then we used these pairwise similarity matrices, we combined them to find the train test pairs that were most similar in terms of all 3 metrics, and then also compared their binding affinity label that is used to train these models. And then we found plenty of structures like this with test complexes with the protein in gray and the ligand in red, and train complexes with the protein in blue and the ligand in green that are basically the same. So you have identical ligands, identical proteins, same affinity, and just a tiny deviation in the ligand positioning. And then this would be another example that is almost the same, or we even found just straight duplicates, just complexes between like pairs in training and testing, where I needed to shift like the structure of the ligand of one that you would even see that there are 2. So we had to conclude that there is like a train test data leakage between this PDB bind database and the CASF benchmark, with about 600 training complexes being nearly identical to a test complex in terms of these 4 metrics. So almost half of the benchmark datasets do not present a challenge to models trained on the PDB bind database. Nevertheless, almost all machine learning-based scoring functions are trained in this setup in the recent years, using the CASF benchmark to measure the generalization and compare to other models. And you can see the published performances of these models here with an RMSE between the true and the predicted binding affinity. And we then retrained 2 models. So this PAFNAcy, a 3D CNN, a really old one, from 2018, I think, and this GenScore, a GNN. And we found that all models in this list are like outperformed by the oldest model when it is retrained on the newest data. So we used the 3D CNN. It actually turned out to be the best in this setup. And then one model is also outperformed by simple lookup algorithms that we devised that just for each test complex search for the 5 most similar training complexes and average their affinity label to make a prediction for the test complex. This performance here. And then we also made a search algorithm that just searches for the training complexes with the 5 most similar ligands and also averages their labels. And this performance you can see here. And then some models are outperformed by a model that is trained only on ligands. So you take the training data, but you remove all protein information, and then the performance is around here. So these models clearly do not really have an understanding of protein-ligand interactions. So we then filtered this training data set to remove this train test data leakage and see how it looks after that. So we just filtered the training data set until all test complexes contained like unique ligands and were structurally distinct from all training complexes. This removed about 600 training complexes, and retraining the state-of-the-art models on this 4% smaller training data set caused quite considerable drops in this benchmark performance. So this GenScore dropped to around this level here. And the previously best one, this PAFNAcy, dropped to around here, close to these simple lookup algorithms. But of course, this is like not a fair comparison because all these models have been trained and evaluated in this setup. So we would have to retrain all of them. And then probably most of those would also drop after filtering the data set. So that's why this figure doesn't exist. And then another example, which goes more into the direction of integrating domain-specific pre-processing to the data, is this protein thermostability prediction. So the task here is to predict the melting point for proteins given their sequence. So you want to know at what temperature a protein degrades and loses its 3D structure, because it's desirable, for example, in biocatalysis, to have enzymes that work at high temperatures, because this can like accelerate your biocatalytic reaction rates and improve the process efficiency. And so the models that try to predict this, they take sequences and then try to predict a melting temperature for this protein. And the training data for this is frequently used this Meltome atlas, which contains about 48,000 protein sequences from 13 species and the associated melting points. And then what some models did is they took these IDs that are in this data set and fetched the corresponding sequences from the UniProt database. And the problem here is that UniProt contains complete amino acid sequences as they come from genetic sequencing experiments. So they also include regions that are cleaved off during post-translational processing. So you might have your genetic sequence, and then if you would fold this sequence, it would result in a structure that looks about like this. It includes like signaling peptides, so extensions that are only very briefly attached to the protein and are cleaved off under like normal conditions, resulting into the mature protein. So it is not really a good idea to take these sequences and train a model on it, because protein stability is a property of the mature protein and not of this protein sequence as it is in the sequence resulting from the genetic sequencing experiments, because this is not a structure that exists under physiological conditions and is very unstable. A third example is molecular docking or co-folding. So the task here is to predict the binding conformation of a small molecule to a protein. And classical docking algorithms are simulation-based. So the protein is looked at as static, and then you simulate different binding conformations, and a scoring function predicts the binding free energies of these binding poses. And then you try to find like the location of binding and the preferred orientation that results in the lowest energy, and this is then the final docking pose. And the co-folding models that we looked at last week, they are usually diffusion models, and they predict the protein structure and the ligand binding pose simultaneously. So they are trained on all molecular complexes in the protein data bank, including small molecules and DNAs and metal ions. So for example, you could give it the sequence of this protein and the SMILES code of this inhibitor here, and then it would just predict the structure of the complex immediately. And examples for these co-folding models would be AlphaFold 3 or Rosetta Fold All Atom, or this recent JAI model and the bolts. And if you look at the protein folding, they all have very good performance that is quite similar to AlphaFold 2. And then the second part of the task is this ligand positioning that it does at the same time, right? And these co-folding models were shown to outperform the classical physics-based docking algorithms. So this is this redocking task. You just take a lot of proteins that have small molecules bound, and you remove the small molecule and see if these models can rediscover this native binding pose. And then there have been other studies that actually showed that these models can interpolate quite robustly, but struggle to extrapolate to inputs that they haven't seen during training. And they also showed that artifacts such as like steric clashes occur quite frequently, so overlap of two atoms, and also that the models are not really sensitive to adversarial challenges that you pose to them. So the conclusion was kind of that these models primarily rely on like data-driven pattern recognition and not on an understanding of the physics of protein-ligand interaction. And I would like to show you that using the results of one paper, using this MOP kinase system. So you have this protein, this MEK1 bound to an inhibitor. And the first challenge that you would give to a co-folding model is just remove the ligand and see if it can rediscover this binding pose. And you see for these 4 co-folding models with the protein residues, so the amino acids of the protein in blue and the original ligand position as it is in the crystal structure. So the ground truth is in gray, and in green you can see the pose prediction of these co-folding models. So you can see that most of these models in this specific system here are able to rediscover the native binding pose of this small molecule to that MEK1 protein. But then they made this challenge of binding site removal. So they mutated all the binding residues to glycine, which is really like unreactive. So basically they removed all possibility for interaction of the ligand with the protein. And you can see here that most of the co-folding model still predicts this native binding structure, although all possibility for non-covalent interaction with the protein has basically been erased by only having glycine in the binding pocket. Yes?
So what would be expected to happen there was not predicting the original line?
Yeah, actually, if the model has an understanding of like how protein-ligand interactions work, then you would have expected, or we hoped to see some ligand displacement so that it doesn't predict this original binding pose anymore, but it just predicts the ligand kind of a bit outside of the pocket so that you see that it has kind of reacted to the presence of these unreactive glycines in the pocket and hasn't really put the ligand into the pocket. And then the next challenge they did was this binding site packing. So it's kind of similar to the previous challenge with adding a lot of unreactive amino acids to the pocket. But here they also mutated them to phenylalanine. So basically removing all interaction possibilities, but also occupying the pocket with really bulky hydrophobic groups. And then you can also see here that most of the co-folding models still predict this native binding pose. And there are cases of like unphysical overlapping atoms and large steric clashes because the binding pocket is basically already full. So the results that I showed you are only based on about 2 protein ligand systems. But they still show that these models, they have a tendency of unphysical predictions. And they show only a partial capacity to adapt to such disruptions. And so they kind of concluded that co-folding models are not predicting poses based on the physics of interactions, but rather just learn patterns in the global protein structures and sequences in the training data. So we need kind of traditional better benchmarks, because traditional benchmarks primarily measure how well these models capture structural data. And we also should think about how we can better integrate like physics into these models, like basic scientific principles. So to summarize this part, some of the challenges that people face when using protein or biological data for model training is that they sometimes have a lack of understanding of the biological context. So they kind of fail to incorporate domain-specific preprocessing, which can lead to models learning irrelevant or misleading features. An example for that would be the thermostability prediction using all the signaling peptides in the sequence. And then the dataset redundancy is a big problem because random and temporal splits, they are insufficient because there are so many intrinsic redundancies. And these usually lead to test sets that are not independent of the training data. And then there's also a lack of physical or chemical constraints in these models. So the models tend to reproduce patterns that they observe during training, even when the results are unphysical. So we should more often validate model outputs against established scientific principles. And we need to think about how we integrate these chemical and physical priors into such tools. And in general, we just need better data sets that are based on a careful curation and data splitting strategy, and benchmarks that like quantify the generalization and also also test the adherence to fundamental physical principles. Okay, so then I will now transition to this de novo design of small molecules. So, and for this, I would like to remember or refresh this diffusion model principles that you've probably seen in the previous lectures. So these probabilistic diffusion models, they achieve state-of-the-art performance in image generation. And this works in a way that they kind of add increasing amounts of Gaussian noise over a certain number of time steps. And then at intermediate time steps, you might still have some images that have a clear resemblance to your input image. Then if you add more time steps at some point, you have basically erased all the information from that image. And then you just train a neural network to undo like these steps of noise. And if you're successful at doing this, you can basically sample from your known Gaussian distribution. So basically sample random noise, feed this to your models, and then retrieve a realistic image that resembles your input distribution, like you would get another small dog, for example. And diff-SPDD is one example of a model that applies these principles for a small molecule design. So it's also like a denoising diffusion model that generates new ligands, but conditioned on protein pockets. So it aims to generate small molecule ligands that bind to like a desired protein structure. And it considers the protein pocket as a fixed context during the diffusion process. And the ligands that this model generates, they are quite novel and really diverse. And they usually match the shape of the protein binding pocket quite well. And the idea behind all this is that traditional drug screening is kind of limited to the chemical space of previously studied molecules. And the hope is that diffusion models can go beyond this and generate entirely new molecules, kind of explore some new regions of chemistry. And to train this model, they just used all the protein ligand structures in a data set called the crosstalk data set, and then added increasing amounts of noise to these molecules within the binding pocket. And then the model is trained to recover this ground truth molecular structure from the noised inputs. And then if they are successful, they can sample through iterative denoising of random noise within the pocket some new structures here, new molecules within the binding pocket. And for this, we need to know how we can add like Gaussian noise to a small molecule that is bound to a protein. And what they do is they first represent this ligand as an atomic point cloud. So just with 3D coordinates x and the categorical features h, which represent the atom types. And all the molecular bonds are neglected. So that is this step number 1 here. You remove all the bonds and you represent this molecule as a point cloud. And then you add noise to obtain this noised structure. So you basically noise the coordinates and the atom types at the same time. And this kind of transforms this atomic point cloud here from aligned with a molecular structure to just randomly distributed. And then they train the neural network to rediscover this structure. The backbone of this model, so the neural network that removes these noising steps, is a GNN. And so these noised atomic point clouds are represented as fully connected graphs. And then they are merged with a protein graph, so the surrounding amino acids within the pocket, but these are not noised. So then the GNN computes updates of the ligand features H and the coordinates X that are needed to reverse one noising step. And the features and coordinates of the protein graph, so the surrounding amino acids, they remain unchanged during this reverse diffusion process. So the workflow works like this, that you have this, in the sampling, you have your pocket, and then you can basically sample a random noise within this protein pocket. And then over this certain number of time steps, you go from time t to 0, basically. So aiming to match your data distribution again. So you end up with a point cloud that might look like this. And then they just add bonds to this point cloud based on like rules, how like a set of rules that defines like how long bonds are between two atoms. So for example, a CC bond has a certain length, and if it's a double bond, then it's a slightly closer, a slightly shorter bond. Yeah. Do you have any questions about that? Otherwise, as I mentioned last time, if I like show you a model, it might not necessarily be because this is the newest and best model there is. So again, for those of you who are interested, there are like a lot of models for like generative ligand design conditioned on the target pocket of a protein. So target diff would be quite similar to the one that you've seen now, but it generally results in a bit higher quality ligands. And then drug flow and flow root, they are flow matching implementations of the same thing. So also the pocket conditioned ligand design, but drug flow has this very interesting concept of using virtual node type to allow the model to dynamically remove atoms. Okay. Then I will transition to the de novo design of proteins and show you two models that are very often used for this task. So everything that we've seen before now related to proteins was basically protein engineering, where you explore the protein space by just modifying an existing proteins to optimize its properties. So the resulting proteins usually share a lot of their structure and also their sequence with the protein that served as a starting point. And in de novo protein design, this is not the case. So the idea is that you generate new sequences of proteins from scratch. And this does not rely on any natural proteins, but uses computational methods to generate sequences that might potentially be very different to all sequences that have been characterized before. So there's this statement that evolution or nature has only explored a tiny subset of all potential proteins that could exist. A famous model that generates de novo proteins is this RF diffusion model. And it's also applying diffusion to proteins to generate realistic protein backbones de novo. So you can think of, it's really similar to what we've seen before. You generate training data by noising structures of the protein data bank. So you take protein structures and add noise until you reach this distribution. And then the model is trained to recover the ground truth structures of these noised inputs. And again, you can generate then new structures through iterative denoising of sampled random noise. So we have to talk about how we can now add Gaussian noise to a protein backbone, because the model operates not on entire proteins, but on protein backbones. And more specifically on the frame-based representations of the protein backbone. And these frames are these triangles that I talked about that all amino acids have in common, of these N, C alpha, and C atoms, which form a nearly planar rigid triangle. And these triangles are connected to each other to form this backbone chain of the protein, right? And you can basically describe each of these triangles with a translation and a rotation. So they model the protein backbone as a set of L translations and rotations. And then they add 3D Gaussian noise to the translations. So they basically perturb the C alpha coordinates. And then they apply Brownian motion also to the manifold of rotation matrices. So the noising process basically transforms these sets of triangles from very nicely aligned along a protein backbone, as you can see in here, into randomly distributed and randomly oriented. And then the neural network is trained to realign these triangles into a realistic protein backbone. Some very interesting aspect about this model is that in this case, like in contrast to all the previous diffusion models that we've seen, the neural network is not trained to predict one denoising step. So it is trained in a different way. So this is basically the neural network here. And it always takes the current coordinates of these frames, of these triangles and their orientations, and then always directly predicts, makes a prediction for the ground truth structure of this protein at this x0 hat here. So it always predicts from the coordinates it gets the structure of the protein that it thinks is most likely. And then, given this neural network output, the input to the next denoising step is not this prediction of the ground truth structure, but a noisy interpolation towards this prediction. So each step basically gets a current set of coordinates, which can be this noisy interpolation towards the prediction of the previous time step. And it gets always, besides these coordinates, also the ground truth prediction of the previous time step. So it is not discarded, but you always take some set of coordinates and the prediction of the ground truth that the last time step, the last diffusion step made into the next diffusion step. So this is what they call self-conditioning. And so the model can use the positions of the coordinates at the current time step and the predicted ground truth coordinates of the previous time step. And according to the authors, this improved the performance of RF diffusion quite a lot. And another reason for this model to be trained in this way, always predicting the ground truth structure of the protein, is that it's actually based on a protein folding model. So it's based on Rosetta fold, which is similar to AlphaFold. So a model that takes a protein sequence and predicts its 3D structure, and it's with similar accuracy than AlphaFold actually. And the idea behind this is that we can achieve improved diffusion models for protein design if we leverage the understanding of protein structures that is already contained in these folding models. So RF diffusion was developed through fine-tuning this protein folding model. So basically taking the weights that can already fold a protein from its sequence and fine-tuning it on the diffusion task. So if you look at Rosetta fold, this folding model, it's quite similar to AlphaFold. You have this input sequence. So the sequence that should be folded into a protein. And then you have also some homologous templates. So it looks for structures of similar proteins that might already exist and makes a first guess of these pairwise residue distance matrix. And then it always uses this frame-based representations of the protein backbones. So it always includes also this first guess of the coordinates of the protein backbone. So if this is the architecture of Rosetta Fold, they basically used the same architecture for RF diffusion. But now the sequence input is masked because in this task we don't need this anymore, right? And then this homologous templates and the first guess of the pairwise distance matrix is replaced by the ground truth coordinates of the previous time step. And the first guess that Rosetta Fold makes on the frame-based representation of the protein backbone is replaced by the coordinate prediction of the current time step. And then the model is trained in this way to always predict the ground truth structure of the protein. Starting from random noise, it can generate protein backbones that are highly diverse. You can see some examples here on the right side. And many of the unconstrained designs that this model generates show very little similarity to existing proteins, kind of indicating that this model can generalize beyond its training data. And also experimental characterization of generated proteins show that most of these are expressible, they are stable and soluble. Another interesting aspect of this model is that it can be conditioned, it can be customized to specific design challenges, for example. So by adding external potentials. And this is kind of like a guiding of the denoising trajectory at each step towards a specific design objective. So the authors of RF diffusion showed this by conditioning RF diffusion with the structure of a TIM barrel protein, a protein that has this overall topology that you can see here. And then they could generate diverse designs that had this rough overall topology. Another thing they showed in their paper is that they can build scaffolds to hold a desired protein motif. So I quickly have to explain this because it's not that simple. So P53 is a famous, it's a tumor suppressor. So it's present in all our cells. And whenever the cell turns cancerous, P53 is supposed to initiate cell death. So it's supposed to keep tumors from growing. But if tumors grow anyway, then they usually have some mechanism of evading this P53 associated cell death. So some mechanism that keeps P53 from exerting its function and initiating cell death. And one way that cancer cells often do this is by overexpression of MDM2 protein, which is a protein that binds to P53 and therefore keeps kind of P53 from doing its job. So cell death is not initiated anymore because all P53 is bound to this overexpressed MDM2 protein. And the design task that they took on now in this paper is to design a competitor protein that binds to MDM2. So kind of like a fake P53 that would bind to all the overexpressed MDM2 proteins so that P53 would again be free to do its job and initiate cell death once it's needed. So they conditioned RF diffusion to include this helix of P53 and then generated some designs that included this helix. And they showed that these designs actually bound to this MDM2 protein quite strongly. And this is kind of like a potential cancer drug. Was this understandable? Okay, good. A bit complicated. Yes, but so far, I've told you that RF diffusion generates protein backbones. So you might wonder, because the protein backbone is not at all a protein yet, right? So it's just the backbone. So something that you might see here, all these amino acid backbone triangles in a chain, but they all don't have anything attached that they're C alpha atoms. So they don't have side chains and they don't have amino acid identities. So what is needed to complete the protein design is to identify amino acids that will, an amino acid sequence that will correctly fold into our desired design. So in our desired backbone architecture. So this is the sequence design task. So you could design a protein backbone, and then you need something to design a sequence for this backbone. And then you can basically take this sequence that you designed, give it to AlphaFold, for example, and check if this generated sequence actually folds into this backbone architecture that you desire. So a model, a potential model that can solve this task is a protein MPNN. And it's a generative model for sequence design that integrates also graph-based representations of the protein backbone. So it follows this sequence from structure objective. Design a sequence for a given fixed backbone, which is also referred to as the inverse folding problem. So you might have a protein like this, and you represent it as a backbone. So only the backbone triangles of each amino acid without side chains. And then you want to find a sequence that will fold into this structure. So you basically have to equip all these backbones, basically all these C alpha atoms, with some side chains, so that once you produce these proteins, the spontaneous process of folding will basically guided by the interactions between these designed amino acid identities and between these side chains, fold the protein exactly in this structure that you desire. So, and for this, in protein MPNN, they model this backbone as a graph, and the nodes of the graph are amino acids in this fixed structure. So more specifically, the C alpha atoms in this backbone. And then they just connect all these C alpha atoms to their 32 nearest neighbors. So through a graph edge, sorry. So it's basically some contact map. So that could more or less look like this. So you have all these nodes that represent C alpha atoms, and then you connect them, each node to the 32 nearest other nodes. And then you also need to add some data, or you can add some data to your node features and your edge features. So, and how they do this in protein MPNN is the node features are just initialized as zeros. And the edge features are initialized as all distances between the backbone atoms of two amino acids to kind of encode the spatial orientation of this amino acid. So remember that each edge connects to C alpha atoms. So kind of like here, between this C alpha atom and this, and then how you would initialize, or how they initialize the edge features. So the features of this edge is all distances that you can compute between the three backbone atoms of these two amino acids that these C alpha atoms represent. And then if you have this graph here, then this very densely connected graph with these features, then they have an encoder GNN, so a message passing neural network, where these initial node features, which are just zeros, and these edge features, which are these distances, are transformed with graph convolutions and message passing steps. And in this way, information about the spatial environment of each of these amino acids should be like integrated or embedded into the node features and edge features at a certain location. And then once you have these updated node embeddings and edge embeddings, they use this graph with these updated representations for the second part of the model. So this is basically what I've just explained. You have this GNN setup where you take your edge features, the distances, and the node features, which are these zeros at the beginning. And then you have this alternating sequence of edge and node updates. So they first like make a node update with an information flow from the edges, and then they update also the edge features with an information flow of the node features, and this is repeated 3 times. So this graph convolution generates new edge and node embeddings, and these should encode some information about the spatial neighborhood of each amino acid. And then the second part is a transformer model that does autoregressive decomposition. So it's going through the sequence. So at each part of the sequence, you want to identify a good amino acid, right? So it's going through the sequence, one amino acid at a time. So this model, the second part, is run as many times as there are amino acids in the sequence. And the input to the model is these updated edge and node embeddings and the previously predicted sequence. And then it returns for this position in the amino acid chain a categorical probability distribution from which the next amino acid is sampled. Good. So if that is clear, then I will continue with the training of this model. So the training data is a data set of proteins with known sequence and experimentally solved structures. So, and then they take all these protein structures and kind of remove all the amino acid side chains. They reduce them to only the backbone, and then protein MPNN is trained to rediscover these sequences that were previously in this protein. No, this amino acids, I'm sorry. And the loss then is just a categorical cross entropy per amino acid. And if they train the model like this, the sequence recovery reaches around 50%. So of the original sequence of the protein MPNN can rediscover about 50% of all amino acids correctly. And you might think that this is really low or kind of low, but we have to consider that at many positions in a protein, more than one amino acid might fit. So there can be more possibilities given the structural constraints at the given location, especially in the outside of the protein. So the more important thing to do is this structure validation. So to find out if these predicted sequences that you've designed for your backbone actually fold into this backbone. So the workflow is like this, that you have this input structure, like this, and experimentally solve the crystal structure of a protein, and then you reduce it to the backbone and remove all side chains. And then you have this protein MPNN that designs a new sequence for your backbone, and you pass this new sequence to AlphaFold, which predicts the structure of this sequence. And then you can check if the output structure closely matches your input structure or not. So I have a small demonstration here. So this is the workflow that I've just shown you, and I will now quickly demonstrate how we can redesign a sequence for a given backbone. So I will choose this MDM2 protein that I've explained before, that is overexpressed in many cancer cells. And this is in the protein data bank under this with this ID. So I can retrieve it like this. And then we have some settings here. So I can say I want to design four new sequences for this protein. And there's also a sampling temperature. So if we increased this, then the model would be kind of more creative about its selection of amino acids. But we'll leave this at 0.1 right now, and I will run this model. So, and you can see here now, it has generated 4 new sequences. And all of these sequences here, you can see, have a sequence recovery of about between 40 and 45%. So all these sequences share only 45% of their sequence with the input MDM2 protein that we've used. But this doesn't tell us if this model has worked, right? Because it's just a sequence. So we use this structure validation approach where we can then see if the sequence has the same structure once it's folded as this MDM2 protein that we used. So I will here run AlphaFold on all these sequences, and this might take a while. So I will go back to the presentation for a moment. So protein MPN solves this sequence design problem. So finding a sequence that folds into a specific backbone. And the experimental validation of the generated structures show quite high accuracy. And this graph-based modeling of the 3D backbone and the message passing derived node and edge features are crucial for this model's performance in rediscovering or in choosing amino acids that fit at the specific location. And an interesting and important aspect is also that also here desired motifs can be integrated by keeping a part of the sequence fixed during the sequence generation process. So if we go back to this MDM2 example, we have designed this protein here that carries the helix of p53 so that our protein then can bind to this MDM2. And now, because we want our generated protein to still bind to MDM2, right? It might be a good idea to use the same amino acids here that like the original amino acids in p53, because we know that MDM2 binds to p53 and we want to keep it away from p53. So we just use the amino acids of p53 here. And then we can like fix this part of the sequence. And then protein MPN will just design the sequence around this fixed motif. So now this has finished. We can see here that we have, for each of the 4 sequences, we have some scores and then the recovery, and also this PLDDT score, which is basically a per residue deviation from the structure. And we can now look, for example, at the one that is the best one, sequence 1, and you can see that if you compare the newly generated sequence, once it's folded with AlphaFold, to the structure of MDM2 that you can see in gray, then you see that this really closely matches the input structure that we used. So we have been successful now in generating some sequences that if we fold them, or if we would produce them in a lab, then it would have the same structure as this MDM2 protein. Let's go back to the presentation. So diff-SPDD that we've seen is a diffusion model that generates new small molecules, new small molecule binders for specific protein binding pockets. And the combination of RF diffusion and the protein MPN that we have just seen is one potential approach for de novo design of proteins. I'm saying one potential approach because there are many more models. This is a really competitive field, and you can hardly keep up with all the studies that are published in protein design. But this is one very good combination of models that is frequently used. It's publicly available, and they work really well. But this has in no way been an exhaustive lecture on this field. So, and the very interesting and important aspect is also that these models can be conditioned to include desired motifs or have desired topologies. So this allows the design of proteins for specific applications. Good. If there are no other questions, then I'm at the end.